Kevin, I'm online. If you want, I can put up my slides.
I think you can take screen control away, and display.
My slide visible?
Yes, it is.
Okay.
Kevin,
Yes,
yeah, there is a general question in Slido.
I did. I saw it. I'm going
You
to
saw
save it.
that.
Yeah, I'm going to
Okay,
save
you
it
saved
for
that.
mine
So-
because I'm afraid I'm not going to get any questions. So,
Well, maybe yours. That's what I want to say. If you have no other questions, we can use that one.
yeah, I'm going to save that one for mine.
Okay. Okay.
All right, welcome back, everybody. We are at the 11:10 Central mark. It's my pleasure to introduce Madhav Marathe from UVA, who will be speaking with us to us from the Biocomplexity Institute that just initiated at our university in the past few years. Madhav?
Thanks. Thank you very much, Kevin, Aidong, and Vipin, and other organizers for inviting me. I'm going to talk about computational epidemiology and in particular real-time computational epidemiology. I don't think it needs much of an introduction given the events in the last few months. This is a team, and I want to thank the team. The work is certainly not my work by any means. It's a work of a large group of folks as well as our funding agencies. So just before I start, here are some references. I believe the slides are going to go online, and folks who have further interest can take a look at some of these papers. So I'm going to focus on COVID-19 because I think you couldn't find a better example at this point of time for real-time computational epidemiology. And but our group has done work on this area for a while now, and the slides and the papers can talk a little bit more about it as we go along. So COVID-19, for those of you who did not follow from the very early period of the pandemic, we got some early cases, or at least some sniff of it in December. And then by 14th of October, you started getting a much stronger signal that this might become a larger pandemic than we had anticipated. And by mid-March, things were fairly clear that this is not going to abate and just stay in China, basically, and it's going to move all around the world in a fairly significant way. And why did this become pandemic? As it happens with other diseases, this is a novel virus as far as humans are concerned, so everyone is susceptible. There are very interesting modes of transmission for this virus, including the fact that folks can be transmitting the disease but might be asymptomatic. And finally, where it started and how it started is also important. It started in a very dense part of the world. Not only that, it also started in a part of the world which is very well connected to the world in terms of airline traffic, in terms of other forms of human mobility. In contrast, Ebola, which is also quite deadly, started in Western Africa, and it's not so well connected to the rest of the world. So it was relatively easier to control it at the start. The social and political response has been amazing. We've probably lost more than $3 or $4 trillion by now. Just US has invested over $3 trillion in this. And you've seen a slew of papers. So people have reported between 6,000 and 20,000 scientific articles and preprints on this topic. So it's just everything about this whole pandemic is quite unique in a certain sense. So we at the Institute have been working on this general problem for a long time now. We've been doing epidemic modeling for 20 plus years, I would say closer to 25 years now. And we have supported the US government as well as state and local governments and international authorities throughout the last 20 years on larger pandemics. We participate in the H5N1 scare. We then participated and actively played a role in the 2009 H1N1 pandemic. We were also part of a large team that supported the DoD and other federal authorities during the 2014 Ebola outbreak, and then finally the Zika outbreak. So we started tracking this in December, and the work formally began in January, I would say somewhere middle of January. And about 80 of us, including staff, students, and full-time scientists, work on this. These are folks here at UVA, but also our collaborating institution. It's a very massive effort. We directly support policy analytics. For the DoD, we provide input to CDC through various mechanisms. We are the primary modeling group for our state and work with the Virginia Department of Health and Virginia Department of Emergency Management. And we also provide science-based evidence and analysis of policies for our universities and local hospital systems in the state. And as you'll see as we go along, this has led to substantial advances in this particular topic of computational epidemiology. So what we would like to do in the long run is what I call P-cubed analytics, basically. And this applies not just to epidemiology, but massive biosocial systems. We want to build systems that are pervasive. We want to build systems that have ability to do personalized decision-making. And we want this to be precise. And there are a couple of interesting papers. There's a nice article in Science during the H1N1 outbreak by Feinberg and his colleague Wilson, which was titled Epidemic Science in Real Time, very beautifully done. There's also an article by Dirk Helbing in general for network systems that I would encourage folks to take a look at, and talks about systemic risk and how system models would be useful. I want to point out the NSF just awarded us a very large project called the Expeditions Project to study this further. And we hope to continue this work for the next 5 years as we go along. So we are not the first to do modeling for epidemics, of course. This whole thing started in 1700 with Bernoulli's work for smallpox. And in the last 100 plus years, the class of model that has been very useful is called the compartmental mass action models. It's a fairly simple and straightforward model, but very, very useful. You take the population and divide into, in this particular example here, 3 compartments, but 3 is just a number. You could divide it into more compartments if needed. S stands for susceptible, I stands for infected, and R stands for recovered. You can again have S there so that you become susceptible again, as it happens in certain diseases. And the basic idea is to write a set of differential equations, which are basically mass action models, and tell you how the rate of density of susceptible infected boxes or compartments would change as as the compartments interact. And from this simple model, which is the workhorse for epidemiology and very, very valuable, you can get important quantities like when the disease peaks, the time to take off, the peak value, the total number of people who might get infected. 2 big quantities that everybody must have read by now is herd immunity, which is getting a lot of attention, and I'll have more to say later on, maybe in a couple of weeks. And also the notion of R0, or effective reproductive number, both of which are important quantities that epidemiologists have studied and are used to guide their decisions. So as much as these models have been useful, there is a certain amount of shortcomings, or shall I say, something left to be desired, which is that they don't capture the heterogeneity of the system, right? And the approach that I would like to outline today is one that I'm calling it networked epidemiology, to studying epidemic processes on multi-scale social contact networks. And the idea and the hypothesis is that a realistic representation of the underlying network and the behavioral adaptation yields better insights into the contagion dynamics. And this basic idea of a representation theory was outlined in Professor Shehu's talk today as well, and also in Tandy's talk in the morning. So in some ways, this is not new. It's just applied to a kind of problem that we are working on. So the basic idea is very, very simple again. You have, just like the models that I talked about in ODE case, 3 states. A node can be, so you take a graph, each node can be in one of the 3 states, susceptible, infected, recovered. The edge labels denote the transmission probability. And in this case, the node red is initially infected, tries to infect V1 and V3 and V2 independently with probability half. And this shows you the time sequence of what happens. So V0 ends up infecting V1 and V2, but not V3. And then V1 and V2 infect V4 and V6. And then finally, V6 infects V3. And then the epidemic stops in this case. So you can write down a sequence of configurations that the system goes through, which is very nicely captured by a Markov chain, where you have, in this case, a node where you start it, so shown in the middle, and then you have edges going from one configuration to another, and the label on the edge is a probability that you transition from one of these super nodes, so to say, or configuration node to another configuration. So in that sense, it doesn't look very different as far as computer scientists are concerned, what one would have when you represent a probabilistic Turing machine, almost identical. As it happens there, the problem lies in the fact that this Markov chain is exponentially larger than the network itself. So even if each node has 3 states, a chain like this would have, and there are n nodes, you would get a chain of size 3 to the n. And this is just a start of the complexity of the problem. So, and most of the analysis problem is not to be done on the base network at all, but on the Markov chain itself. So an output of this effort is called a dendrogram. And a dendrogram represents how the disease progress. In this case, V0 is going to infect V1, V2, and V2 infected V4, V6, and V6 infected V3. And this is shown in a tree-like form in the bottom right corner of the screen, and picks up the basic transmission in a certain sense. So think of this as stain. People do this in neuroscience where they put a stain and then look at which part of neurons light up. There's not conceptually something similar is happening here. You have a stain and it stains certain nodes, and this tells you how the nodes get stained. Provably, the problem is actually very hard. It's number P hard, and so we start to use simulations to try and understand these systems. The problem is what I've talked about till now is about epidemic simulation, how the disease spread. The problem becomes even harder when we start talking about interventions. And one of the most common interventions that all of you have now read endlessly in newspapers is one of social distancing. That's the only way you can control COVID-19 right now. And the basic idea, you want to reduce social contacts to try and reduce the spread while also minimizing the economic and social impact. So here's an example of what a social distancing would mean in our toy example that I showed you before. So let's start from left top and then go around. So the first issue is, how do you intervene? So when do you intervene and for how long do you intervene? So in this case, V0, V1, and V2, you're going to intervene, so take them off, and you're going to lift the intervention in this case at time t plus delta. The second thing on the top says, who do you intervene and how do you intervene? So you can either close workplaces in this case, or isolate the nodes or quarantine the nodes. And isolation quarantine is different. In case of isolation, you isolate the folks who are sick. Quarantine denotes isolating individuals who have come in social proximity of the infected individuals, but they might not be sick themselves. And then you talk about the pulsing strategy, where you close the place for a while and social distance for a while, then you open and then you social distance again. This is a strategy that was discussed in Neil Ferguson's paper as well, and others have studied, but I would not have ever thought that this would be taken seriously because it's very, very expensive to do it. But you can see that a version of this is really playing out as we try and control the COVID pandemic. So as I've argued, hopefully so far, it's building computer models and simulations really are the only way to solve realistic versions of this problem. And in the next 5 or 10 minutes, I'm going to tell you how we go about doing this. There's a lot of theoretical work as well. I've tried to skip that. The papers and the tutorials that I have referenced early on has all the details that you would like to look at. So the way we go about doing this, and which I think is novel in a certain sense, is to do this study of epidemic processes or networks in 3 steps. So if you want to study how the disease is spreading in, say, Washington, D.C. or Charlottesville, then the first step is to build a digital twin of that city. And to do that, you combine various information that is thrown at you, census, roadways, building locations, travel diaries, and build out a digital representation of it. And what you get from this is a network. And the intended meaning of this digital twin is that you have a representation of the city or the region that you're interested in that is statistically very similar to the region you want to study. But none of the agents have any sense of direct one-to-one correspondence with individuals living in the city. And the papers will make this very clear of what statistical similarity means. I won't go through it here. But effectively, such a system provides a social coordinate system for understanding how cities are built out. So here's a cartoon of how we do it. We take census information, we take travel diaries, we take road information, we take digital traces, we take flow data from various devices as whenever we get handle on, and build out essentially what we call a people location bipartite graph, which is on the right side of your screen. On the left part of that bipartite graph are individuals with lots of labels, and on the right side are locations with labels. And effectively, a person, if he visits a location L, the person has an edge from person node to the location node, and the edge also has a number of different attributes. So this is the graph you end up generating. This is the social contact network. It's a people location graph from which you can do a projection on the people side to get a people-people interaction graph. And that is the network that we are talking about, and we will use throughout our study in doing this. So this is a very interesting big data problem. It's been a while. We've been working at this, as I said, close to 25 years. And there's no real stopping criteria in the sense that these things get better and better as new data sets come in. We have now built out such a digital representation for the entire US, and we will release versions of it in time. So the second step is to build agent-based models that understand how diseases spread on these networks. It turns out that you need HPC-like systems to do anything meaningful with this because they're fairly complicated. And here is an example of how disease manifests itself in a single person. I showed you a simple model, which was SI and R. In the real world, it turns out the models are much, much more complicated. So here's a model we use for a single individual when we study COVID. It has got about 15 states and captures a lot of interesting structural aspects about asymptomatic and symptomatic. When do they need medical attention? When can they get hospitalized? How can they recover? And all of this information is picked up. This is for folks who have done formal work. It can be best cast as a probabilistic time transition system. So transition in this probabilistic automata are both timed and are probabilistic. And you have one such automata for every individual in the population, right? And the transitions in this automata are dependent on the interaction that the individuals have with each other. So here's a simulation that we performed some time back with Sanjay at UIUC and his student Abhinav and a number of other folks at Livermore in our group. And this was actually a non-trivial task where we could simulate epidemic spread. This time it was influenza through the entire US using the cluster at Lawrence Berkeley Lab. Prabhat was very helpful in getting us those machines. And we could run this in about 8 to 9 seconds on 800,000 plus scores. And this is needed if you start doing analytics with it. So the third step is to build workflows because that's really what is needed to finally do decision support. So you want to solve using these workflows questions like forecasting, analyzing different interventions, doing economic impact, studying inverse problems like finding out patient zero, inferring various transmission probabilities, computing the risk of individuals as well as a collection of groups, and looking at the attack rate. So what we have done over the last 4 months for COVID is we have built out a number of different dashboards to support decision makers. One that is quite popular and probably second to the Johns Hopkins dashboard is a surveillance dashboard that we built early on, very similar in its form and content, but a few different features than Johns Hopkins dashboard that many of you, I'm sure, know. We built out another dashboard for our state to tell them how the medical resources are going to be used in the state in the next few weeks. The basic dashboard is generalizable to US. We are working on the models to drive it, but it tells hospital systems in the state when they can expect the bed capacities to be overshot if the disease continues on the path that it is on. On the left, below, we have produced what's called contact matrices. A lot of epidemiologists have interest in it that tell you how individuals interact with each other, and you can build these social contact networks from it. And the fourth one at the bottom is a joint effort with Stanford, Michael Bernstein's group, and Jacob is, I believe, leading that effort there. But we're building a crowdsourced platform to collect various social distancing measures counties in US are putting together. And this has been challenging, actually. We thought we could quickly do this at the peak of the epidemic in March, April, in about a month, and we are still far away. It's not straightforward to get this data. It's not straightforward to make sure it's correct and update it all the time. We've also built decision support systems, and I will say a little bit about it. On the top left, we did a study, and this is on medRxiv, it's under review. We studied early on how would the disease make its way to other countries once it starts to move out of China, and we could do a reasonably good job on predicting this, what's called importation risk. On the right side is our weekly projection for our state. We do a fairly detailed analysis, and this data is available on state website every week after we are done. So you can find today's update that we gave them just now in an hour back on VDH website by Monday or so. And this tells you every week where the state's sort of numbers are and where they're likely to go in the next 2 to 4 weeks. We have steadfastly avoided making long-term projections because I personally think they're meaningless, and I'll say a little bit about why. This is a question, though, everybody's interested in, from news reporters to policymakers to scientists. The problem is that unlike physical systems or even systems such as weather, where the error in the forecast is largely because, in a physical system like predicting the movement of moon or other planets, the error is very limited. In case of weather, you can argue that most of the problems arise because we don't have absolutely accurate representation of the model, or at least not enough time to solve those equations with the precision that we want. But we understand the physics quite well, we would argue. Here, the problem, and this is true for every socioeconomic system, is that predictions become endogenous to the system. So our predictions, and even without predictions, people's perception of the disease and their risk-seeking abilities define what they would act in the next few weeks. So a prediction that shows the numbers to go skyrocket upwards might never turn out to be true, and oftentimes do not, because most of the time these predictions lead to people taking preventive behavioral measures that change their behavior, and then the numbers come down. This happens all the time in such systems, and so it becomes non-trivial to forecasts which are way ahead. You can do forecasts for a couple of weeks with reasonably good accuracy, at least in some cases, if the data starts becoming available. At the bottom left, we have a paper with Google now using flow data that we got from Google. This is flow data that Google collects. It's anonymized aggregate data based on digital devices and tells you what flows exist between any two patches in the world, and these patches are roughly 2 square kilometers big. So if you take a patch IJ in this global grid and you have another patch KL, then this tells you the flow and the human mobility flow, normalized and aggregated, anonymized, but still has a lot of information in a certain sense. And the plot will show you how these flows are able to capture global change in mobility around March 15th or so. And something interesting happened. The government orders came around 15th of March and a little later in most places. But the events in Italy and the events that were starting to unfold in New York actually had people change their behavior even slightly earlier. So this basically tries to tell you how policies can impact individual behavior, or especially their own perception of risk would impact their behavior. The paper actually goes further and does retrospective analysis of what would have happened if you had delayed these lockdowns, as they're called. So I'll quickly spend time, last few minutes, maybe.
Madhav, just to warn you, you're at time through the questions.
So that's very good. So let's skip this. We have a Bayesian ensemble. Let me just summarize, as Kevin said. What I want to let you know is real-time epidemiology is indeed possible, and the role of models should go beyond predictions and structured understanding. And finally, social, economic, and political dimensions are actually becoming increasingly important as we understand epidemiology. So this is not just a problem of virus spreading between two individuals, but hugely affected by these considerations. So I'll stop there, Kevin, and take questions. Thanks.
Excellent, thank you. I saw one question on Slido related to earlier in your talk about some of the Markov chain analyses and things. You gave a nice example of how quarantine might be encoded isolation. And the question related to how would you encode masking, facial coverings and things, because would you randomly reduce probabilities through the network, or are there other strategies for that?
It's a very good question. So like all the other talks you've heard so far, data becomes a real challenge. So now there is some information on masks, but if you look at the survey that New York Times and other agencies did recently on mask coverings, you still get an aggregate number, right? And this number, of course, changes in time. So the model provides you with the flexibility of deciding how to put it in, but the way you represent it finally will affect the dynamics. So you end up trying to do the best you can with the data you have. And in this particular case, we would take certain average values, hopefully broken down by age groups, which is very, very important. We know that it matters. And spatially as well, you know that certain parts are much more compliant in the US than other parts, at least that's what the numbers suggest. And then to use the best and then do uncertainty quantification over it. We are doing a similar analysis for campus reopening, just for folks who might be interested, and we'll release a report in the next 2 days. It's actually quite interesting what might happen as campuses start to reopen or not.
All right, excellent. Thanks very much.
Thanks.
Our next speaker is me. Let me just get my slides set up here. Okay, wanted to start off by saying what a great pleasure it's been to co-organize and now attend this workshop on knowledge-guided machine learning. As a Packard Fellow, I used to attend annual meetings where we would hear these terrific talks from all areas of science and engineering, chemistry, physics, astronomy, mathematics. And it drove home to me the point that I most enjoy conferences and symposia that bring together diverse fields, like what we've been witnessing over these past few days. It also taught me that I needed to and how to raise my game when speaking to diverse audiences. And so if there are any hydrologists, climatologists, or any aquatic scientists still left on the call, I think that there were a couple last I looked on the participant list, this talk's for you. I'm going to introduce one project in translational biology from my group, which ends where I think that knowledge-guided machine learning could pick up. I'm going to intentionally tread lightly on the details because I personally find that big acronym soup to be somewhat off-putting. And this publication here on the bottom sorry. Here we go. Provides all the experiments, analyses, and mathematical details. All right, my focus today is on oxidative stress and its precursor reactive oxygen species. And I'll begin with what these terms mean chemically and biologically. Reactive oxygen species arise when oxygen reacts with an unpaired electron to form a highly reactive molecule called superoxide. This is what bleaches a fluorescent molecule if you stare at it for too long, and a downstream derivative of it, hydrogen peroxide, is a component of all those hand sanitizers that we're using by the hour. Why are reactive oxygen species antimicrobial? Well, it's because they react rapidly with all shapes and sizes of biomolecules. And these chemical modifications are often detrimental to the organism. And more on that to come in a moment. But before I get there, I want to tell you where these free electrons and by extension reactive oxygen species come from within cells. And the answer is mitochondria. This is a subcellular structure that powers all nucleated cells through what's called an electron transport chain. Hopefully you see where this is headed. The electron transport chain itself is quite good, but the initial transfer of electrons from the rest of the cell to the mitochondria is leaky or lossy, meaning that mitochondria occasionally fumble the handoff of an electron from the cell and allow it to escape, react with ambient oxygen, and create superoxide. Mitochondria get even clumsier when, instead of being fused together as a contiguous network, think a lava lamp analogy, they're broken up into little pieces through a process called fission. And this fission is required when cells divide. Since cancer is characterized by uncontrolled cell division and proliferation, oxidative stress is important for the disease. However, its precise role is extremely complicated. On the one hand, superoxide can directly damage DNA, give rise to mutations. Thus, the motivation for consuming antioxidants in dark-colored vegetables, or for some, taking the highly concentrated pills available on the nutraceutical shelf of the grocery store. However, reactive oxygen species also disrupt other biomolecules that, if that damage is in excess, would be detrimental both to microbes, to cells, and even to cancer cells. Thus, one could argue that reactive oxygen species serve a tumor suppressive role, and there's evidence for antioxidants waking up tumors that would otherwise be dormant in the face of excess oxidative stress. Unsurprisingly, our cells have evolved elaborate mechanisms for sustaining life in the face of oxidative stress. From the standpoint of detoxification, our cells express a suite of enzymes that take reactive oxygen species and convert them to progressively less reactive products, culminating in the formation of water. More interesting, to me at least, is that reactive oxygen species also serve a signaling function, acting as second messengers that turn off families of enzymes, which normally keep signaling pathways inactive. Inhibiting an inhibitor is equivalent to activation. Of note for this talk are the ubiquitin ligases, which tag specific proteins for destruction within the cell. If these ligases are inhibited, the targeted protein accumulates and then can go on and exert its function. We were examining two such proteins, which serve as transcription factors regulating the expression of several of these detoxifying enzymes, along with a number of other important cell stress effectors. NRF2 is a transcription factor stabilized by oxidative stress, as well as electrophiles derived from broccoli sprouts, and has a two-phase role in cancer, with moderate activity thought to suppress cancer, but chronic stabilization thought to promote it. P53 is another transcription factor, but it, by contrast, is nicknamed the guardian of the genome and is the most commonly mutated gene in human cancers. When we stained for the accumulation of these two proteins in non-cancerous breast epithelial cells, shown in the lower left here, we observed a sporadic stabilization that often, but not always, coincided in the same cells. To orient you, the scale bar here is 20 microns, and the images that you're looking at are cross-section, multicolor images of maybe 24 cells. We quantified the NRF2 P53 co-stabilization across hundreds of cells and analyzed their relationship by mutual information, which asked, given the stabilization of one protein, how much does that reduce uncertainty about whether the other will be stabilized? And what we found was a significant statistical coupling, non-zero mutual information between these two proteins. Why would these two proteins be stabilized in the first place? Well, we were observing them expand in a three-dimensional culture format that requires a good bit of cell proliferation and reorganization under conditions that generate a lot of stress. So much so that some of the cells in these 3D cultured spheres die because of the excess oxidative stress. At this point, the translational biologist should be asking, who cares about a pattern that arises from what is possibly a cell culture artifact? But what I can tell you is that the pattern is disrupted when we perturb these cells genetically, and we find that 3D growth and organization is altered in non-obvious ways when we do this disruption of these transcription factors and disruption of the pattern. We also observe similar patterns in the mammary glands of mice during puberty, as well as in normal breast tissue from women, shown in the upper panels here. Interestingly, we found that the coupling was statistically elevated in breast premalignancies, these so-called ductal carcinoma in situ, or DCIS lesions, both in clinical material, as shown on the right here. This is a DCIS lesion with the margin I'm highlighting with the pointer, as well as in cultures of DCIS-like cell lines. Recall that the mutual information in the former slide was about half that shown here. Strikingly, in breast cancer, this coupling breaks down entirely. Had multiple instances of single positive cells found in the tumors. So something here is engaged, activated during premalignancy, and then blown up during the course of the disease. To organize our observations and evaluate their consistency with known mechanisms, we turn to systems modeling of the established physicochemistry. In the opening session, Marcus Reichstein flashed a couple of examples of systems modeling in the geosciences, and I want to emphasize the distinction here for good systems biology. In the schema on the left-hand side here, there's not an influence map of dependencies that have been documented or postulated. It's as close to fact as biologists can achieve. In other words, I know that if I take purified keep-1 and mix it together with hydrogen peroxide, that it will be directly oxidized, keep-1 by the hydrogen peroxide or its downstream products. Furthermore, if I take this oxidized keep-1 and mix it together with NRF2 under the right conditions, it will no longer be able to act as a ubiquitin ligase that degrades NRF2. And for the math aficionados, I'll say that these models are not much more than mass action kinetics, coded as a system of delay differential equations with rate parameters estimated either from timescales of the biology or from direct biochemical measurements done in the lab. Before this work, we had good physicochemical models for the NRF2 and P53 pathways, citations here in magenta and green, but no one had previously found a reason to join the two together, which is what we did by carefully considering, and for the most part excluding, instances of crosstalk between the two modules. We then used the integrated model to simulate our experiments. In the NRF2 P53 stabilizations observed, if they came from bursts of oxidative stress, and those bursts came from mitochondrial fragmentation as a result of cell division or other triggers in the culture or in the tissues, then we would expect these bursts to arise randomly and asynchronously, such that any fixed snapshot of a culture or a tumor or a tissue would reflect the cells adapting at different times to the oxidative perturbation. Using the model, it was straightforward to parallelize our simulations and gather random snapshots at random bursts of oxidative stress, and then evaluate things like mutual information that resulted from those simulations. When we did this, we observed relatively good concordance with the statistical couplings documented in the 3D cultures, including a doubling of the mutual information observed going from a non-cancerous breast epithelial line to a pre-malignant DCIS-like cell line. Overall, the relationships are a lot stronger, but that's because the model is devoid of any statistical blur caused by experimental noise, which is considerable for the experiments that we were doing. As importantly, the model could make predictions about synergistic changes in overall time integrated oxidative stress when one factor, NRF2 or P53 or the other, was perturbed genetically or pharmacologically. And these likewise agreed with the emergence of phenotypes, not showing here, that were observed when NRF2 or P53 were jointly disrupted in experiments. We sought to leverage these capabilities in the realm now of cancer data science. One of the advantages of working with cell lines is that all the information is out there in the public domain these days. Accessing genetic data on hundreds of lines through the Cancer Cell Line Encyclopedia is straightforward. We extracted 15 breast cancer transcriptomes, that's all that acronym soup on the top here, that describe the abundance of 20,000 or so genes expressed by these cells, and mined out the 15 species that were direct surrogates in the model. Another set of acronyms over on the right here. Then we used these line-to-line variations in the abundance to define different initial conditions for the model, which were then iterated as before to calculate things such as mutual information and overall oxidative stress handling. And what we found was that the cancer cells with high oxidative stress tolerance, this is reactive oxygen species, coincided with high mutual information, a high statistical coupling between NRF2 and P53. Among these cells, when we genetically perturbed the NRF2 pathway, we actually didn't see much of a phenotype or an effect when we grew them in 3D. Conversely, on the other end of the spectrum over here, there were lines with low tolerance, low mutual information, and growth phenotypes when we blocked NRF2 signaling genetically. The rub was that the phenotypes were not all in the same direction. Some lines grew worse, other lines grew better with NRF2 pathway disruption, and there would be opportunities for machine learning to discriminate between these two groups if we could dramatically increase the number of lines that we perform this analysis with, perhaps by subcloning out different derivatives of these lines that are shown on the top. Where it got really confusing was when we pivoted to using the exact same approach with transcriptomic information, not from breast cancer cell lines, but instead from clinical breast cancer cases from the Cancer Genome Atlas, or TCGA. On the left, this was a slide from before, compressed version on the left-hand panel here in H, are the expression profiles for the lines. And what is clear is that the breast tumors are very different in their gene expression profiles, same genes here along on the rows. Some genes are expressed at entirely different abundances from what was observed in the culture. as an example, MDM2 here on the top row. Whereas other genes are much more spread about the average culture abundance. A good example of that are these detoxifying enzymes shown in the 3 rows here. Further, when we ran the simulations, the nice relationship between oxidative stress tolerance and NRF2 P53 mutual information broke down, including several notable cases of low tolerance, but extremely high coupling between P53 and NRF2. They're highlighted by the tick marks there in the middle. In these cases, perturbations of NRF2, envision the woman who dutifully takes her concentrated broccoli sprout supplement, could have a dramatic impact on tumor growth. We just don't know right now which direction that impact will be. Will it be a positive or a negative impact? And so in closing, I want to conclude with where I think knowledge-guided machine learning could help us in the next steps of the puzzle. To review, what we have is a physicochemical model that takes in gene abundances, passes them through a biochemical network, and makes predictions about stress coupling and handling. Moreover, what I can tell you is that straightforward regression of genes to predictions fares terribly. So that there's something in the model that the model is doing beyond a straight mapping of abundances to incremental changes in prediction. But we can't figure out what that is. Perhaps some type of neural network architecture, probably with a relatively small number of nodes and hidden layers, could be useful to us if the inputs and outputs are constrained by the topology of the network. For example, negative feedbacks would be a constrained instance of backpropagation. In this way, the oxidative stress dynamics act like a convolution of sorts, kernel, relating the things that we can measure to the things that we care about, with the machine learning providing insight into how the model got there. The advantage with this type of formalism is that we can simulate as much synthetic data as needed in the range of abundances documented for human breast cancers to guide the learning. At this point, it's just an idea, but it's one that was really nucleated as being part of this HDR group that you've been hearing about over the past several days. And I'll conclude with my acknowledgements. This work was led by a recent PhD graduate from my group, Liz Pereira, who really regrets not being able to present this work to you herself. She was aided by multiple undergrads, graduate students, and research associates in the group at specific facets of the project. The experimental work was supported by the National Institute of Health, the grant numbers shown here, and a lot of the computation was conceptualized and driven forward as a result of the HDR support. And so with that, I will thank you and take any questions from the audience.
Hi, Kevin. Just one question. You mentioned how the machine learning will help in the future work. And also, I'd like to know a little bit detail what kind of prior knowledge really will help in your research.
Okay, and so the first one was, yeah, so I got hung up on the second one, and so I didn't quite hear the first one. Could you repeat it again? The first part of the question.
Oh, I just hope to, I saw you slides for the future work and machine learning. So how the prior knowledge, what kind of prior knowledge will help?
Oh, yes. Well, I think that this topology, so the connections here, if there's a neural network type of architecture that is constrained, some of the edges don't have absolute flexibility to be gained and lost and amplified or decreased. If there's a rigid structure at certain places, but then a machine learning type of flexibility to weight edges elsewhere in places where we want to have non-linearities and we want to have things be emphasized and de-emphasized. For us, I think that would be very valuable because we can still look at those weightings through this prior knowledge about which factors interact with which other factors. And in the models, in these simulations, we keep track of all of these species. and we can measure them over time, the dynamics. Now, our end readout here are the integrated results of many time-dependent simulations. So it's not as straightforward as a time series, and we want to auto-encode and go things from there. But it does provide us access to a rich source of observations to look at how those things should be weighted. But I think having a little bit of rigidity by the biological knowledge, it would be beneficial for us for interpretability, because we really want to look at the weighting through the pathways that we have this understanding about.
Okay, thank you. I no more questions, I think.
Oh, but I will take, so I will take the, how are we doing for time? We're we're at time. Oh, okay, we're at time. So then we'll go on. All right, so then I will exit here, stop my share, and introduce you. So our last speaker of this session is Aidong Zhang from University of Virginia.
Yeah, let me share my screen. Do you see it?
I can see it, not at full screen, and I still see your presenter view.
Oh, okay. Oh, okay.
That's the one.
Okay, everything good? Okay, so hi everyone, last talk of the session. And so I will talk about how the newest kind of machine learning called meta learning can be used for cancer prediction. So in general, we have seen lately deep learning applied to bioinformatics. So majority of research actually are in the multi-omics for cancer prediction, because omics data have been generated a lot lately. We also see biomedical image analysis a lot, and also literature mining, such as what Kathy just presented, and DNA sequence function prediction, protein interaction prediction, and RNA protein binding size prediction. So all these different predictions have been investigated lately. But we are seeing the limitations of applying deep learning into bioinformatics because of the very well-known problem called the big P, small n problem, which means we only, although we have a lot of data, we only have a small number of samples in each cohort compared with the high dimensional features each sample has. Unlike the pixels in the images, like very successful applied in Google competition in images, the patient features such as gene expressions are much noisier and also more heterogeneous. These features are not, don't have orders and very high dimensional. So how to deal with this in machine learning context? So for deep learning meets biomedicine and health, so our goal is to design novel models that can make an accurate prediction based on much less training data. So that's our goal in applying deep learning to the bioinformatics. And also we want to develop customized deep learning models that can incorporate domain knowledge to make more accurate predictions. With that, I hope to just mention, we recently published a paper in AAAI, top AI conference last year. The title is Semi-Supervised Deep Learning for Cancer Prediction. So in that method, we designed a neural network which utilizes the both supervised and unsupervised way by utilizing the relationship between the inherent gene relationship and enforcing the KNN inside of deep learning. So it can, we design multiple layers in deep learning with KNN models, so we can quickly converge to the better neural network for prediction. So if you are interested in that, please look at this paper. So, but beyond that, I want to talk about more recent machine learning new trends in machine learning, which make me excited how this can be applied to cancer prediction. So what is a new trend in machine learning? So currently the research under supervised learning paradigm is using large and data and models to generate generalizing the model and for the prediction. However, you don't, as I mentioned, you don't have that large data set to train this deep learning model. So, so machine learning community is looking at how humans really learn. So we have, so if you look at the kids, so kids, how they learn, they have, so for example, they see cats and birds just a few times, they can quickly tell them apart, right? So, and people, you can learn how to ride a bike and normally can quickly learn how to ride a motorcycle. So that capability, you know, it's not reflected in machine learning these days. So machine learning is using a huge amount of data to train the data, to train the model. So can we actually train the model with few data and make a better prediction? So that's what the machine learning community is struggling with. And basically we have all this, you know, long tail problem, which you have a lot of small data and how you will train your model. So with that, people lately propose this called meta-learning. So the meta-learning is to mimic, try to mimic human learning by training the machine with a variety of different learning tasks, which has only few samples. And so you want to utilize that and to be able to make predictions using the few samples in the testing set. And so the meta-learning definition more specifically is to learning how to learn. And the good meta-learning model property is the goal is to adapting or generalizing to new tasks and new environment. In general, there are two stages in the meta-learning. The first stage is the basic learner trained for training tasks and update after each task. And second stage is the meta optimization, so updated by the testing task. So I would say generally meta-learning also has a feature of this transfer learning, how to transfer the knowledge between data sets. But it's more advanced than the transfer learning. How is the transfer? So this memo is a famous model proposed by UC Berkeley in Europe's paper. So if without going to the technical detail, just look at this picture. So you are training a matter part, this is theta, the parameters. So this is the global parameters you are training. And then you will be able to use this quickly adapt to different tasks later when you have the testing case. So that's generally the meta-learning using the gradient model called memo. And so if we more visible, in a more visible way, you have the different tasks, and each task is small. So here, the top left one is two-way, it's called two-way 4 shots. So it's in each task, and you only have 2 different classes, and each class you only have 4 shots, which have 4 samples. And using these 4 samples, and with the query or testing case, and you will train the model. So you will have thousands of such small tasks and testing case to train your model to be able to easily adapt with a small number of samples. So that's what the meta-learning is trying to do. And more generally, for example, this one is 5-way 10 shots. And so using that to train the model. So the reason I mentioned this new machine learning technique is I'm excited, want to see how this can be applied to bioinformatics, especially, I'm going to skip that page, especially if I assume everybody is familiar with the TCGA, the Cancer Genome Atlas. So this famous data set has around 20,000 samples with 33 different cancer types. But if you look at this table, in each cancer type, you only have like a few hundred samples, okay? So if you want to train your model, use each individual sample set and make a prediction for each individual. So it's normally very difficult because the size of sample is too small. You cannot really train a deep learning model with this. Then you will want to use the whole data samples. But we will show using the whole data samples with different approach, compared with the meta-learning approach, meta-learning can do much better than all the other traditional approaches. So the TCGA dataset has all different kind of features. So you can see methylation, RNA, different kind of expression and protein extension. We have been using all these data features to test our model. So basically the goal for the cancer prediction using TCGA is we want to predict the classification of cancer. And the idea is to take advantage of label the patient from different type of cancers and design the learning model to be able to adapt quickly to the specific cancer you want to predict. So it's a kind of knowledge transfer in the sense if you want to think about knowledge transfer or transfer learning. So the steps in this process using the meta-learning is you have sampling and you have meta training with each small tasks and for each cancer, and you have inner training and meta training, and then make a prediction. And so we have set up using the TCGA data, and we use the 31 different cancers, and I think 33, 2 of them are too little, we drop those. And then we training classes, we have randomly selected 21 cancers. And then validation, we use the 5 cancers. And then the testing classes, we use the random selected 5 cancers. And we use the random sampling applied. So I hope to show you how the latest result we just received. So using 3, so this is the result we got. And you can see we have 3 ways and 1 shot, just using 1 shot in each way, each class. And we compare with the KNN, random forest, matching network, prototypical network, and memo-based. So the memo-based came the top and it performed extremely well compared with the other method. And as you have more shots, like 5 shots, you can understand 5 shots give more examples in each class, so it performs better in this. And when you mix all the samples using the traditional way, some of them working well, some of them, especially you see the 1 shot for Canaan, it does not work well. but if you have 3 cells or 5 cells, it works much better. So this experiments make us very excited how we can use meta-learning for cancer prediction. So with that, we started to further looking at, how the prior knowledge can be used for the prediction. So we want to examine different kind of knowledges. So we look at the prior knowledge in the bioinformatics domain. So you can combine this knowledge in the graph point of view. So for example, for image, you can organize into hierarchy categories, different hierarchy. And for patient data, you can organize into different diseases. And so that is very useful for utilizing the domain knowledge. So with that, we first experimented with Mimic, which is the disease data, very famous disease data with the ICD-9 code, which is more diverse than the cancer, the TCGA data. And so we have, so the reason we started with the Mimic data is the disease, they have the code. With different disease code, 3-digit code and 4-digit code. With this code, you actually can well organize them into different categories. This kind of prior knowledge can be used into the machine learning model and to make a better prediction. So we used these graphs and experimented our approach. And this actually is going to be presented in KDD conference is happening next week. So we use the Mimic data with the graph, and you can see this and our approach at the bottom. So this performance is much lower than the TCGA because the Mimic data is more diverse. So we experiment with a subgraph sampling, means we only sample the tasks from the subgraph defined this way. And you can see the subgraph performance is lower than if random sampling, because if you restrict two subgraph and they are more homogeneous, so it's more difficult to detect, to distinguish different diseases. So random sampling actually perform better. So so that's what in the Mimic data sets we perform, and we compared with all different kind of latest approaches without prior knowledge. And we show in this chart with prior knowledge, we can achieve better performance in general. So that is Mimic data. Now, ongoing research, what we really wanted to do is to still go back to the cancer to predict the survival outcome and analyzing the gene set across different cancers. So we also want to perform interpretation of the predictions. So in the survival analysis, there is a difficulty because you have censored data, which you don't know when the time specific event occur. So we combine with Cox, famous Cox model into the survival analysis. So that is a predefined process. Now our ongoing research here, more difficult part compared with Mimic data is how to combine the cancers into a prior knowledge graph. So we don't have, there is no such predefined graph to categorize all different cancers. By collaborating with Virginia Medical School faculty, we lately, so we have started to combine different cancers into graph by calculating the expression data. So if you use the expression data, then you can build different graphs. So in cancer, there is no unique defined graph like the mimic disease ICD-9 code. So you have you will have different graphs built by different expressions, protein expression, gene expression. We'll build different graphs to be used for cancer. So that's what we are doing right now. So I don't have the result yet on this graph. So basically the meta-learning based approach for the genomic survival analysis. So I think my time is up. So generally we have our goal is to build this meta-learning based approach for survival analysis. And we're going to use TCGA with all different samples and 33 different cancers. And so there are 78% of censored data to be used in this research. And we are going to build different graphs based on the prior defined cancer categories using different features and see how that works. So that's a general ongoing research we are doing. So I want to conclude my talk here. So there is still a long way to go, I believe, and there are many interesting problems in bioinformatics and health informatics for which new machine learning methods can help. And this research requires very close collaboration between computer scientists and domain experts, so the machine learning method can well incorporate the domain knowledge into the prediction model. Okay, so I will stop here.
All right, thanks very much, Aidong. We had
I'm on time.
Yeah, there's a couple, right on time, right on time. We have a couple of questions, one somewhat specific, and then I'll also throw that general one to you at the end. But first, we'll start with the specific one. I had a question about the types of biological data that would make them good candidates for the few-shot learning applications that you described before. Biological data is very heterogeneous. Your applications were very sequencing based, but there are also image data sets. There's other types of biological resources that are available. Just want to know your thoughts on which ones you think would be good candidates for that.
Oh, well, that's a very good question. So in general, I think all different data sets fit into this model, and the imaging data actually have more bioimaging data, and imaging data fit well with this because it has, it's proven the image data can, in general, can make a very good prediction using the meta-learning. So as I said, the problem is with different cohort. Each cohort has very few data. So I think the meta-learning will fit well with the cancer prediction or other diseases. But we need to be able to design advanced approach, how the research, the new research here, I think, is how to incorporate the different biological knowledge into this model. That's something new to us to make a good advanced model for machine learning. So that fit well with the theme of this workshop, knowledge-guided machine learning, right? Mm-hmm. So I think it's exciting how we can incorporate the different knowledge into this model.
Mm-hmm. Yeah, the one example that comes to mind outside of the realm of cancer is this effort called the Human Protein Atlas, which is trying to localize every protein within the cell. And I know that there are certain cellular sub-localizations that aren't occupied by many proteins. but something is targeting them there, something presumably in the sequence. So you could envision a learning where based on sequence information with only a few examples that localize to some rare subcellular localization, you might be able to attempt some type of learning like this. So, okay, let me ask I'm gonna ask a good closing question here that got thumbs up. We'll save it to the end. General question for any of the session speakers, which means you. What does translational biology need? Does it mean making data usable in many different disciplines or something more specific? So what does it mean to you?
Maybe any speakers want to take it first? Anybody want to take it first?
For me, when I think of translation biology, I think of RNA to protein. Maybe, but the other thing is this kind of flexibility to it. I guess you could say, from my standpoint, some of the things that I talked about, basic science to more bedside clinical applications, that type of bench to bedside translation. But I think that there's enough, it's a big enough tent that many definitions could fall underneath it. I do like the idea of use and reuse for data for different applications beyond the ones that the original data creators had intended. That's supposed to be the long-term view and the long-term value of these data repositories, that people will come up with creative ways to deploy them beyond what the original intention was.
Yeah, to follow up that question, I think, this translational biology can be very well used in drug purposing and compound design, because you have, different drug, the compounds you want to design, and they normally have fewer samples, or you have prior knowledge a lot of prior knowledge you can apply to the process.
Marta, do you have something to say? Yeah, I was going to say, Marta, we'll turn the video on. Does she have something to add?
Yeah, I wanted to give a sort of a different perspective on it. It doesn't mean that we are here to replace, or that we claim to replace, the experimentation, the chemists, the biologists, or the physicians, but we reduce their hypothesis space. We point them to a narrower set of credible hypotheses, so that then they can take the ball running and get to the right answer much faster than if we were not in the picture.
And I think that that's one of the main points of this workshop, is that a straight ML application doesn't get you there. It doesn't give it, from the experimental standpoint, none of that gives rise to the types of hypotheses that we would really be able to test in the lab, but perhaps by this hybrid approach. So what, Kathy, you had something to add?
Yeah, and I think that really is the take home for this workshop, right? Brings together domain scientists and machine learning experts, computer scientists, to really do this in a very collaborative, interdisciplinary fashion. And I think one thing that is really exciting about this initiative is that the framework is supposed to be adaptable, whether we are looking at the environment science or health sciences or any other application domains. And so, yeah, I think it would be really exciting to see what come out from the discussion regarding the type of reusable module. You were talking about the FAIR principle, right? The reusability, interoperability. And so oftentimes we talk about that's, how you codify the data so that it will be reusable. But in terms of the models, the machine learning models, how could we design in such a way that it could be applied to various data types, and as long as we have good understanding of the feature space, I think that would be extremely exciting.
Excellent. Well, I think that that's about as good of a way to end this session, because it's a great transition to the closing panel that we'll be having in just about an hour's time. And so we'll close the translational biology session. Thank you for everyone who participated. And please do circle back to us with the closing session at 1:30 Central, where Kathy and several other individuals who you haven't heard speak at this workshop up to this point will be participating in the panel discussion moderated by Emma and Paul. See you then.
I just have one housekeeping item before we break. If you are planning on joining that session at 1:30, please do refresh your connections with either the Zoom link or the YouTube link that you used to access this session this morning. We will be redirecting those links during the lunch hour.
Okay, great. Thanks.
Yeah. Thanks, everybody. Thanks, Amanda, Kathy, and Ted. Amanda, thank you. And
Thank you all.
and
Bye,
Ted.
everyone.